Hello, my name is Philip Alex. I'm a senior at the University of North Florida, and I'm currently finishing my last semester of biomedical science. My senior seminar mentor is Dr. Rahman, and today we're going to look at whether mutations in DAF2 and C. elegans potentially lead to extended lifespans in mammals. Our goal is to determine what causes the longevity effects. We want to establish whether or not these effects are conserved, and if it'd be an efficacious treatment for aging in mammals and in human beings. So mutations in DAF2 equated to longer lifespans in the model organism C. elegans. So C. elegans are ideal because they have a short lifespan of about 2 weeks. They have a small genome of about 100 million base pairs, versus human beings, which have 3 billion. They're very easily reproduced, and also they share key metabolic processes with higher organisms. So this phenomenon was first observed in Cynthia Kenyon's laboratory at the University of California. What they found was wild-type C. elegans had an average lifespan of about 18 days, compared to DAF2 mutants, which have an average lifespan of 42 days. The DAF2 mutants also became full-sized adults. They had normal brood sizes, which meant it didn't affect their fecundity or fertility, which is biologically important. Also, pumping, which is the contractile function of their pharynx, ingesting food, it decreased, but in DAF2 mutants, it prolonged and it persisted even in old age, meaning they were still metabolically active. Well, DAF2 relies on the activity of its primary transcription factor, DAF16. And what they found was when they knocked out DAF16, even if you mutate DAF2, you don't observe any longevity effects. But how does this work? So, mutation of DAF2 genes leads to decreased DAF2 receptors or the ligands that bind to the receptor. Essentially, it leads to nuclear localization of DAF16. And DAF16 upregulates expression of a series of protective genes that enhance proteostasis, oxidative stress resistance, DNA repair, immune function, and stem cell maintenance. Overall, longevity. So DAF2 works via insulin IGF signaling pathway. And as you can see here, it's highly conserved across all organisms. What we want to find out is whether or not disrupting this pathway leads to the same effects in other organisms, and whether or not advancing gene editing technology can lead to potentially combating aging in higher organisms such as mammals and humans. And what's exciting is DAF2 encodes for a receptor that's orthologous to human insulin IGF-1, which is very interesting because that's ultimately the end goal. So why is this significant to us? Well, ever since the discovery of antibiotics and the establishment of sterile medical practice, as you can see here in the early 1900s, we see a 10-year increase, a 20-year increase. Ever since then, we've only seen, over the last 50 years, an 11-year increase in total lifespan for human beings. And this is significant because ultimately we want to extend our lifespans, but not only that, but our health too. So firstly, we'll look into Drosophila melanogaster flies. In this study, they mutated the peptide that binds to the insulin receptor in Drosophila flies, called Drosophila insulin-like peptide 2, which leads to downregulation of Drosophila insulin-like peptide 1, which further induces AKH, or adipokinetic hormone. Now, AKH is a functional homologue of glucagon, which its function is to mobilize lipids and carbohydrates. Essentially, catabolizing fatty acids, triacylglycerides, leading to a fasted state. So now this fasted state is what leads to this longevity, because your cells in turn notice this, and they want to fold proteins better. They want to conserve energy. They have these oxidative stress resistance effect, and we can see that increase here. Now in this study, they mutated the actual insulin receptors, and that led to an 80% increase in lifespan. They also mutated a substrate protein downstream called Chico. It's an insulin receptor substrate protein, which led to about a 40% increase in lifespan. So does the observed longevity work via activation of FOXO transcription factors? And the answer is yes. As you can see here, this is Chico. This is what they mutated in the previous slide. However, DAF16 is considered part of the FOXO family transcription factors. And these set of transcription factors regulate apoptosis, cell cycle progression, oxidative stress resistance, cellular metabolism. So it's very well likely that this works within this mechanism. Now we can move on to mice. As you can see, mutations made in growth hormone receptors in mutated mice led to smaller bodies, but about a 30% increase in lifespan. And in knockout insulin receptor mice, specifically at the adipose tissue, they saw an 18% increase in lifespan. Plus, when they fed the mice a high-fat diet, they didn't get fat. They also have Snell dwarf mice and Amy's dwarf mice, which have pituitary defects that lead to chronically low levels of growth hormone and IGF-1. So as we see here, decreasing the IGF signaling or growth hormone signaling, it might lead to smaller morphological characteristics, but essentially it leads to increased lifespan. Over here, this is a chart of a Snell dwarf mice versus the wild type. And as you see, the decrease in body weight, the mass. However, you have also changes in metabolism. You see a decrease in insulin and glucose, but conversely an increase in adiponectin. Adiponectin leads to glucose and fatty acid oxidation, which also has anti-inflammatory and antioxidant activity, which can explain the longevity effects that we see in Snell dwarf mice. Now, this is interesting and it's very important because there's also a study that have seen that a single IGF-1 polymorphism haplotype is common to small dog breeds. So all small dog breeds have this polymorphism at the IGF-1 and nearly absent in giant dog breeds, suggesting that the sequence variant is a major contributor to body size in small dogs. So this is significant because small dogs have a longer lifespan than big dogs. As you can see here, small dogs have an average lifespan of about 14 to 17 years plus, and big dogs have an 8 to 10 year average. And because they're missing an allele for IGF-1, they're smaller, but they live a lot longer. But does longevity mean that you have to sacrifice size? Absolutely not. That's not the case, and we know this because in C. elegans, the DAF2 mutants live twice as long, but have normal body sizes. The Drosophila fly mutants also grew to normal size when they were treated in young adulthood as opposed to after being hatched. And also in mice defective insulin receptors in the adipose tissue, they grew to normal size. So what is the conclusion? Well, perturbations in the insulin IGF transduction pathway leads to extended longevity. We see this in DAF2 mutations in C. elegans, DILT2 mutations in Drosophila melanogaster, as well as the insulin receptor mutation, growth hormone mutations in mice, and also the haplotype observed in IGF-1 allele in small dogs. So this is interesting, but what we saw previously is a genetic induction of the extended lifespan. Well, what we see here is more of a pharmaceutical drug-induced extension of lifespan. Well, metformin in diabetic patients led to increased lifespan than other patients that took other anti-diabetic drugs. And the mechanism of how this works essentially induces a fasted state. It's almost like a dietary restriction. And it also disrupts the insulin IGF-1 signaling, which leads to that extension of lifespan that we see. And as we know, we're advancing in gene technology, and we have CRISPR-Cas9. And what would be interesting is if we can couple the advancing genetic technology we have now with further research as to the effects of mutating insulin IGF-1 signaling pathway factors, whether or not this can be efficacious for human beings in order to extend our lifespans. So hopefully we can couple these two. Some of these studies were published 10 years ago, and the gene editing technology back then probably aren't as good as the technology we have now, especially with CRISPR-Cas9. So it'd be interesting to couple those two and to see if we can advance this therapeutic way in looking at combating aging and what have you. But thank you very much for your time. And if you have any questions, please let me know. Have a nice rest of your day. Thank you.